Hi all, and welcome to the Stem Cell Technologies webinar entitled Human Pluripotent Stem Cells for the Treatment of Age-Related Macular Degeneration. Today, this might be a bit more interactive seminar than usual, since we will be two presenters talking under the same story. The story of our common research over the last 6 years that aims to pave the path to the clinics using cellular therapy for patients suffering from age-related macular degeneration. My name is Sandra Petrus-Reurer, and I'm a postdoctoral researcher working at the Department of Clinical Neuroscience at Karolinska Institute, under the supervision of Professor Anders Cavanta and Associate Professor Fredrik Lanner.
And my name is Alvaro Plaza Reyes. I recently graduated as a PhD from Fredrik Lanner's lab at Karolinska Institute, where I've been working with both human pluripotent stem cells and pre-implantation human embryos. We hope that we managed to keep your attention and interest in a special cell type of the retina that has a great potential to be used as a replacement therapy, and that we study from several perspectives, which we will try to cover as best as we can in the coming slides. And as Amanda said, we would like to kindly ask you to keep your questions for the end of the talk. Age-related macular degeneration, or AMD, is a retinal disease characterized by the progressive loss of vision in the center of the visual field, as well as by the impairment in high-resolution vision in general. So a patient that is suffering from advanced stages of this disease will experience a vision similar to the one on the right-hand side of the slide. Age-related macular degeneration is a highly prevalent disease, considered the third most common cause of blindness worldwide and the first cause of blindness in developed countries. As its own name indicates, the area of the eye that is affected during AMD is the macula, a relatively small area situated near the center of the retina in humans and in some other mammals. If we zoom in this area, we can find several defined layers integrated by a variety of different specialized cell types. On the top part of the region that sits closer to the vitreous, we find the ganglion cells, a special neuronal cell type that collects all the information from the visual world sensed by the different neurons present in the retina, including amacrine, bipolar, horizontal, and photoreceptor cells, and transmit it to the brain. Then we have one of the most important cell types in the eye, the photoreceptors. There are two types, rods, which are responsible for black and white vision under low illumination, and cones, responsible for high-resolution color vision, that are especially majoritarian in the macula. Underneath the photoreceptors, we have the retinal pigmented epithelium, or RPE, which is a polarized hexagonal and heavily pigmented cell type with important functions in the maintenance of retinal homeostasis. The RPE sits on top of a basement membrane called Bruch's membrane, and together with it conforms what we call the blood retinal barrier that separates the retina from the underlying vasculature or choroid. Generally, AMD is classified in two types, wet or dry, depending on the origin of the retinal impairment. Both types start with emergence of subretinal yellow deposits known as drusen, although they are not believed to be the cause of the disease. Wet AMD is characterized by the abnormal growth of blood vessels that end up invading the retina and disrupting the RPE layer, which will end up dying. Differently, in dry AMD, there is no abnormal growth of blood vessels, and instead RPE just degenerate due to still not completely understood reasons. In both cases, disruption of the RPE layer leads to the subsequent alteration of the retinal homeostasis that end up with the death of the photoreceptors, which is the ultimate cause of vision disability experienced by patients with AMD. While there are treatment options for patients suffering from wet AMD, including the intravitreal administration of anti-VEGF antibodies, there is not yet an efficient treatment available for the dry type, which, on the other hand, accounts for the majority of the AMD cases. The important role of the RPE in maintaining retinal homeostasis and its implication in the degenerative forms of AMD has drawn the attention into the potential of transplanting healthy RPE cells to prevent the secondary loss of photoreceptors and potentially to preserve or restore vision. So, aiming to find a better source of RPE cells for transplantation, some authors have recently begun to explore the possibility of generating RPEs from human pluripotent stem cells. There are two types of human pluripotent stem cells that are currently being used for differentiation into RPE. We can use human embryonic stem cells, which are derived from the undifferentiated inner cell mass of human embryos, or induced pluripotent stem cells, which are derived from adult somatic cells via chemical or genetic reprogramming. Now, bringing a stem cell therapy to the clinic is not a trivial procedure. It requires to go through several hurdles, and most importantly, pass all of them in a successful manner. Hurdles that include having a xeno-free, defined, and functional protocol, that this protocol proves to be robust and scalable, that the RPEs produced are safe and pure, that the final product proves to be immunocompatible for successful transplantation, and finally, that it can be produced in a GMP manner before it can be used in a clinical trial with patients with AMD. So we will now go briefly through the efforts that our lab made in order to overcome each one of these obstacles and get an almost ready out-of-the-shelf RPE product. Our first goal was to establish a xeno-free and defined protocol for the derivation of RPEs from human embryonic stem cells, as the majority of published protocols back then were still relying on animal-derived products and media formulations containing serum, which could lead to future complications. So we established a protocol that was completely xeno-free and defined, and that relied on the spontaneous differentiation of aggregate stem cells after removal of basic FGF and TGF-beta from the media. Briefly, we cultured the stem cells in a xeno-free and defined media until they were confluent. Then we scraped them and let them aggregate in suspension until they formed embryo bodies. These embryo bodies were kept in suspension in a modified version of our pluripotent culture media, where we got removed the basic FGF and TGF-beta. After 3 weeks in culture, embryo bodies started to produce pigmented structures called optic vesicles that were allowed to mature and expand for a couple of weeks more. Then around week 5, we manually dissected out these optic vesicles, enzymatically dissociated into single cell, and placed them on biologically relevant substrates, where they were let to expand and mature for another 4 extra weeks until a homogeneous monolayer of pigmented and hexagonal RPEs was produced. These cells were then dissociated and used in subsequent subretinal transplantations in our animal models. One of the main challenges while developing our protocol was to identify a biological relevant substrate that could best support the expansion and maturation of the RPEs after optic vesicle dissociation. For that reason, we assessed the performance of 4 different human recombinant laminins that are naturally present in the Bruch's membrane, including laminin 111, laminin 332, laminin 511, and laminin 521, and compare them to gelatin, an animal-derived substrate typically used for the expansion of RPE by many other groups. We found that laminin 521 was the best in supporting the homogeneous expansion and maturation of RPE cells, and for that reason, we decided to continue with it. We then performed extensive in vitro characterization of these stem cell-derived RPE cells that were led to mature on laminin 521 and found them to display the typical cobblestone morphology and pigmentation that was inherent to RPE. These cells also proved to express RPE-specific markers at the transcriptional level, such as bestrophin-1, RP65, and PMEL, and at the protein level, such as SO1, CRALP, sodium-potassium ATPase, and bestrophin-1. We also tested the in vitro functionality of these cells by assessing their capacity to phagocyte photoreceptor outer segments, their capacity to form an epithelial barrier, which was measured through transepithelial electrical resistance technique, and the polarized secretion of growth factors such as PEDF, all of them typical functional features of a mature RPE. We compared their functionality to the functionality of undifferentiated stem cells, and according to what we expected, we observed that only mature RPE cells and not the stem cells were able to phagocyte photoreceptor outer segments. The TIR values also showed us that stem cell-derived RPEs were able to form an epithelial barrier that displayed electrical resistance, and ELISA measurements showed us that according to what we expected, the RPE secreted PEDF mainly in the apical compartment, once more demonstrating the functionality of our RPEs. We then wanted to test the performance of our cells in an in vivo context after subretinal transplantation. So for that purpose, we took advantage of the rabbit animal model established by our collaborators at St. Erik's Eye Hospital here in Stockholm. The main advantage of this animal model was his eye size, which was very close to the size of the human eye, allowing us to perform subretinal transplantation and subsequent monitoring with instrumentation identical to the clinical setting. Also, we chose to use specifically albino rabbits since they lack pigmentation in their native RPE, and that facilitated even more the detection and tracking of the pigmented transplanted cells. Once the RPEs were mature, which was around week 9 of differentiation, they were enzymatically dissociated and resuspended in a saline solution. The solution was then injected subretinally via intravitreal injection until more or less 50,000 cells were delivered in the rabbit eyes. We then tracked the integration and survival of the transplanted cells both in vivo through the monitoring of the retinal fundus and also later on through histology. From the retinal fundus and histology pictures, we observed that upon transplantation, our RPEs lost their pigmentation, probably because they underwent some sort of EMT phase, but they regained it a few weeks later. So looking at the retinas 2 to 3 months after transplantation, once the pigmentation was clearly distinguishable, we could confirm that our cells managed to integrate successfully, and what was even more important, they did it in the right location and forming a monolayer. Then we also kept some animals for evaluating the long-term survival of these transplants, and we found that the integrated cells were able to survive up to 8 months, which is the longest we kept the animals. Positive immunostaining for human-specific antigen NUMA confirmed the human origin of the cells, and positive immunostaining for RP65 confirmed their RPE identity. Finally, we went on to assess the in vivo functionality of these transplants. In previous publications, we have described that in albino rabbits, subretinal injection of PBS alone creates an AMD-like phenotype, including the photoreceptor loss. We therefore tested whether transplanted RPEs possess some photoreceptor rescue capacity. For that, we compared the effect of injecting PBS alone or PBS together with our RPE cells on the outer retinal thickness of the rabbits. We observed that the outer nuclear layer, which normally consists of 5 to 6 layers, as you can see here on the control animal, was reduced to a single layer in PBS injected eyes. Whereas eye transplanted with human embryonic stem cell derived RPEs displayed an almost intact outer nuclear layer. We then conclude that our human embryonic stem cell derived RPEs possess in vivo functionality and were able to successfully decelerate retinal damage progression, possibly by rescuing photoreceptors from degeneration. We had now overcome our first hurdle, which was to establish a xeno-free and defined differentiation protocol for the generation of functional stem cell derived RPE cells. However, we were aware that there was still a lot of room for improvement in our protocol, since the initial steps depended on spontaneous EV-based differentiation followed by the manual dissection and digestion of the differentiated optic vesicles. There was still a significant batch-to-batch variability. Also, to date, there has been a lack of useful cell surface markers for the RPE lineage that could facilitate in-process monitoring of differentiation efficiency and enable the automatization of RPE cell enrichment and purification. For those reasons, we embarked ourselves into a new project where we sought to identify novel cell surface markers for RPE and make use of these novel markers to develop a more robust, direct, and a scalable differentiation that doesn't rely on embryo body differentiation and that doesn't require the manual selection and expansion of the differentiated RPEs. In order to find novel extracellular markers for the RPEs, dissociated optic vesicles and mature and expanded RPE cultures were screened by flow cytometry using an antibody panel recognizing 242 different CD antigens. We found CD140B, or also called PDGF receptor beta, to be expressed in both the optic vesicles and mature RPEs, suggesting that it could be a good positive selection marker, as it was able to discriminate the RPEs even during earlier phases of the differentiation. There were other two markers, GT2 and CD184, that were detected in the optic vesicle, but were absent in the mature RPE. Those were selected as potential negative selection markers, assuming that they were probably detecting alternative lineages that emerged during RP differentiation, but that were not present in the latest stages of the protocol. These 3 candidate surface markers were then validated by flow cytometry of dissociated embryo bodies at week 5 of differentiation, and by immunofluorescence staining of mature RPE cultures. And we found that CD140B expression was restricted to the optic vesicle and homogeneously distributed in mature cultures, while GD2 and CD184 were mainly present in the non-pigmented areas of the embryo bodies and were completely absent in mature cultures. So now, seeking to develop a new protocol that does not rely on embryo body formation, we use our recently validated surface markers to identify the best culture conditions for enabling 2D differentiation of human pluripotent stem cells into RPE cells. We test the performance of 2 different laminins present in the Brooks membrane, laminin 111 and laminin 521 as substrates, as well as addition or exclusion of activin A in the culture medium, which was previously reported to be an inducer of the retinal lineage on neuroectodermal cultures. Now, instead of enabling the RPE differentiation to take place in embryo bodies, we force the differentiation to happen on adhering cultures by simply changing the culture media from pluripotent culture media to media without basic FGF or TGF-beta that we supplemented with activin A from day 6 of differentiation. At day 30 of differentiation, these cultures contain a heterogeneous population of RPE cells that were then replated onto laminin 521, where we culture them for another extra 30 days until they produce a homogeneous, pigmented, and mature RPE. We then compare the performance of this 2D differentiation protocol with our previous embryo body-based protocol and found that our new approach was able to yield 10 times more CD140B positive cells, meaning 10 times more RPE cells. And that was over the time course of 7 weeks of differentiation. Moreover, these RPEs that were generated using our novel 2D protocol also displayed co-expression of typical RPE markers with our novel CD140B extracellular marker, as well as morphological feature typical of mature RPEs, like the presence of surface microvilli and the polarized localization of subcellular organelles observed by electron microscopy. Finally, we also demonstrated that the generated RPEs were able to integrate and form a polarized monolayer after subretinal transplantation in our rabbit animal model. We then examined the robustness and reproducibility of our novel RPE differentiation protocol by testing it with 7 different pluripotent stem cell lines. The differentiation efficiency was then assessed at day 30 and day 60 by flow cytometry for our markers and qPCR for RPE-specific gene expression. We found that 6 out of the 7 pluripotent stem cell lines tested managed to differentiate efficiently, demonstrating robust expression of RPE markers at the transcriptional level and fairly homogeneous pigmentation. As an example, here in the slide, I'm showing the differentiation with 3 human embryonic stem cell lines, where only one, HS983A here, proved to be non-responsive to our differentiation protocol. We see that the responsive lines, here H9 and 980 in green and orange, displayed levels higher than 60% of CD140B positive cells by day 30, and these levels increased to close to 100% by day 60. Also, when we look at the negative markers, we saw that the responsive lines showed almost no presence of GD2 or CD184 positive cells by day 60. Contrary, on the non-responsive line, 983A, which showed no expression of typical RPE markers at the transcriptional level, was characterized by very low levels of CD140B positive cells by day 30, and also high levels of GD2 and CD184 positive cells by day 60. Altogether, these results prove that the identified cell surface proteins can serve as quality control to evaluate the efficiency and quality of the RPE differentiations. Now, to assess the potential of our surface markers for RPE cell enrichment, we compared cultures that were either replated or enriched with a combination of CD140B positive selection and negative selection using GD2 or CD184 at day 30. Replated and sorted populations were then allowed them to culture on laminin 521 for 30 more days and were then analyzed using single-cell RNA sequencing. The TSNE plotting of the single-cell RNA sequencing results indicated that the cells analyzed were distributed into 3 clusters with distinct gene signatures. There was a majoritarian RPE cluster, a second cluster with an I-field progenitor gene signature, and a third small cluster with a gene signature characteristic of a mesoderm. We found that while replated cells harbor 11.3% of I-field progenitors and 1.2% of mesoderm contaminant, these proportions were diminished to 3% and 0% when cultures were enriched using our cell surface markers, highlighting the potential of these markers for increasing the purity of the final cell product. Indeed, characterization of FACS-sorted RP cultures demonstrated that cell enrichment using our markers can improve the pigmentation and maturation of the RPE cells, as well as their functional performance, especially during suboptimal RPE differentiations, as shown here by the increase in tier values observed in sorted cells versus replated, and also in the PDF secretion values also that were higher in sorted cells versus the replated ones. So we have now achieved a more robust and scalable differentiation strategy, which combines the use of activin A with the selection and expansion of RPEs by bulk passaging. This protocol has proved to render a cell production yield that is 10 times superior to our previous suspension protocol. And apart from that, we have also identified a set of cell surface markers that could be very useful in monitoring differentiation efficiency and in increasing the purity and homogeneity of the final cell product. Now we are ready to move on to perform preclinical studies to prove the purity and safety of our RPE cell product. And for that, I will let my colleague Sandra take the lead.
Indeed, and hi again, everyone. As Alvaro said, now we are ready to move on the assessment of the safety and cell purity of the derived RPE cells in several directions that actually should be checked out successfully to be able to proceed clinically. This evaluation now includes karyotype check, analysis of mutations acquired during the differentiation at DNA level by whole genome sequencing, analysis of RPE cell purity, and also pluripotent signatures, both at transcriptomic and proteomic levels, in addition to the assessment of the ability of the derived cells to form teratomas in immune-compromised mice or to migrate to other organs when injected subcutaneously in mice or subretinally in the rabbit eye. Now, going a bit more in detail of what we found, first, the derived cells showed to be karyotypically normal and genomically stable when analyzing somatic and mosaic single nucleotide variation, or SNVs, copy number variation, or CNVs, and structural changes at the chromosomic level, including translocation break ends, inversions, insertions, segmental deletions, and tandem duplications. And although some changes were indeed found, we concluded that they were most likely a consequence of timing culture rather than due to the differentiation protocol. And most importantly, none of them was found to be in any cancer driver genes. Secondly, the derived cells did not show any transcriptomic reminiscence of a pluripotent stem cell, as it can be appreciated in these TCNE plots, now showing 2 clear clusters representing the RPE cells here on the top and the human embryonic stem cells here on the bottom. And as you can see, the RPE cluster show negative expression of pluripotent markers and high expression of an RPE signature, then indicative of the high purity of the final RPE product. Thirdly, we also tested the ability of the cells to form teratomas by subcutaneous injection in immune-compromised mice. And as you can see in this graph, the injection of a minimum of 10,000 cells, that is represented here by this blue line, already made detectable teratomas, whereas not even the injection of 10 million RPE cells showed teratoma formation 7 months after injection. Finally, we assessed the migratory potential of the cells to several organs when injected subcutaneously in mice or subretinally in the rabbit eye by detecting human material by qPCR. The organs included lung, spleen, liver, heart, kidney, and gonads for both species, and optic nerve and vitreous for the rabbit. Overall, no human transcripts were detected in any organ, except for some more specifically in the optic nerve and vitreous samples, which most likely was a result of something contamination to the neighboring retina or to cells that could potentially remain in this aqueous material after partial RP reflux that was actually observed during the subretinal injections. Altogether, it was very encouraging to see that the differentiation protocol did not introduce malignant instability and was highly pure, then successfully going through a quite thorough hurdle and also serving as an important methodological guideline for future safety testing of either stem cell-derived RPE cells, but also of other stem cell-derived products that are facing the clinics. So at this point of this obstacle track, we have cells that are derived in a xeno-free, defined, functional, robust, scalable, safe, and pure way. But it's of major importance to mention that these cells will only last days after transplantation if the immunological makeup is not compatible with the one of the patient that is going to receive them. And because of this, cells will then be totally rejected, not feeling welcomed in the recipient's body, and cleared in no time. So for this reason, we thought it was important to invest some extra efforts on making them as invisible as possible for the recipient's immune cells. So with that in mind, we decided to design the following HLA escaping strategy to make immune-compatible human embryonic stem cells. Then, if cells are left as they are as wild-type cells, the derived RPE cells will most likely be recognized by both the innate and the adaptive immune system, call it T cell and NK cells, and then they will be cleared up. So simplistically, the way it works is that a specific T cell receptor will recognize either HLA class 1 or 2 molecules from the injected cells. So CD8 positive T cells will recognize HLA class 1 complex and CD4 positive T cell HLA class 2. And rejection will happen if this HLA molecule is present and mismatched. On the other hand, the native immune system works a little bit different. And so for NK cells to not kill or to not clear up, they need to recognize the HLA class 1 complex because it serves as a flag to tell that that cell is from their own body. So then if that complex is missing or being mismatched, the recognized cell will be targeted to clear up. So as you can see, it's definitely not an easy task to trick our immune system, although it's also true that it seems that is to a big extent HLA dependent. So we then decided to edit the stem cells that will be further differentiated into RPE cells by knocking out HLA molecules using CRISPR-Cas9. So first we targeted beta-2M, which is a key molecule that aids presenting HLA class 1 to the cell surface. And then on top of the beta-2M background, we targeted C2A, which is the master transcriptional regulator for HLA class 2 genes. So like these HLA molecules will not be seen by T cells, and therefore the injected cells will be expected not to be attacked by them. Although the problem might still remain with NK cells that will lack the recognition of HLA class 1 molecule in the knockout cells, and then they might still get activated. So firstly, and very briefly, we derived single knockout beta-2M RPE cells lacking the beta-2M molecule, as you can see here in this immunofluorescence staining compared to wild-type cells. You can also see that HLA class 1 remained present intracellularly, as beta-2M is only key for the surface expression of HLA class 1, but its absence in the surface was actually shown by flow cytometry when compared to wild-type cells. Similarly, we also derived double knockout RPE cells lacking HLA class 1 and 2, and although HLA class 1 was still present intracellularly, as also shown before with the single knockouts, it was not present in the cell surface. And also HLA class 2 was not detected when assessed both by immunofluorescence and also by flow cytometry compared to wild-type cells. Remarkably, it must also be said that the differentiated RPEs from both single knockout beta-2M and double knockout cells did not show any off-target mutations linked to disease or tumorigenicity, and they did not show any irregularity in the differentiation or survival, as they exhibited characteristic morphology and pigmentation of native RPE cells. So altogether, single knockout beta-2M and double knockout RPE cells lacked surface expression of HLA class 1 or both HLA class 1 and 2, but retain RPE morphology and expression of RPE-related markers. We then embarked ourselves in understanding the reactions of the immune cells, both T cells and NK cells, when they get in contact or in co-culture in vitro with the derived and edited RPE cells. So for T cells, we measured the amount of interferon gamma that was released by the cultures as a measure of T cell proliferation and therefore activation. And in these plots, you can see in the x-axis that T cells from two different donors were stimulated with IL-2 and anti-CD28, and also mixed with or without RPE cells and in different RPE T cell ratios. So we could then see that when RPE cells were co-cultured with CD8 positive T cells here on the left, only wild-type cells, represented by the black bar in these plots, that don't lack HLA class 1, made T cells active and proliferative at both 1:20 and 1:50 ratios, whereas the single knockout and double knockout RPE cells kept them inactivated. And when co-cultured with CD4 positive T cells, was only the double knockout cells that now here you can see represented by the gray bar, that managed to significantly dampen down this activation because of the lack of HLA class 2, that is what is now specifically recognized by the CD4 positive T cells. So for NK cell responses, we both assessed NK cell activation or degranulation by CD107 expression by flow and NK cell cytotoxicity by chromium release upon RPE cell killing. So to our surprise, and as you can appreciate also in these graphs, the NK cell behavior was mostly driven by the donor rather than by the cell type, then being independent, it seemed, of the lack of HLA class 1 or both HLA class 1 and 2. This unexpected effect might be due to the nature of the RPE cells and their known immune suppressive character, maybe expressing a specific NK inhibitory ligands that would then compensate for the lack of this cognate HLA class 1. So lastly, when the edited cells were transplanted into the xenographed albino rabbit model subretinally and without suppression, we could see, judging by the infiltration present in these OCT images, that all transplanted eyes with wild-type cells were rejected at 7 days as a consequence of an early rabbit immune response to the human cells. Whereas the eyes transplanted with either single knockout beta-2M or double knockout cells had a significant reduction of the rejection rate. We also assessed the late response by detecting anti-human IgG antibodies present in the rabbit serums at day 7, 14, 30, and 90 after RPE cell injections without suppression. And as you can appreciate in these graphs, antibodies were detected already at day 7 when wild-type cells were injected, as you can see here with the blue bars. And the titer actually got boosted at day 14. Whereas with the edited cells, regardless of lacking HLA class 1 or both HLA class 1 and 2, antibodies appeared 2 weeks later, so at day 14, and maxed out around day 30, therefore demonstrating a significant delay in the host late immune response towards the derived cells, human RPE cells, in this xenograph model. So in conclusion, the edited double knockout RPE-derived cells evaded both CD4 and CD8 positive T cell responses, did not increase NK cell cytotoxicity, and when transplanted in the large eye model without suppression, they reduced the acute inflammation, and they also delayed the appearance of anti-human antibodies. So therefore, passing another hard hurdle in our track for making the derived cells now immune compatible. So with all this in mind, and as a summary, patients with dry AMD suffer from a degenerative disease causing their vision impairment and with no available treatment at the present moment. So the use of cellular therapy is today one of the most promising approaches to control both the progression of the disease, but also to preserve the retinal cell layers that are still healthy. So our group then contributed to this road to the clinics by generating a source of cells that is xeno-free, defined and functional, robust, scalable, but also safe and pure and immune compatible. And also recently we derived the GMP clinical grade pluripotent stem cell line that will be differentiated to RPE cells and used for a future clinical trial in the coming years, with the hope of getting through all obstacles and approach the finish line in the most successful manner, to finally be able to help patients with age-related macular degeneration. And with that, we would like to thank all the people involved in this work, starting from Karolinska Institute colleagues, led by Associate Professor Fredrik Lanner, who were key for the in vitro characterization of the cells, Santeric Hospital colleagues, led by Professor Anders Kevanta, that carried out rapid subretinal injections and OCT imaging, and collaborators, led by Associate Professor Helen Kaipe, Dr. Evren Alitsi, Dr. Anna Falk, and Dr. Emmeline Nandro, for all the help with reagents and the immunological characterization of the derived RPE cells. Also, we would like to thank you all for listening, and please don't hesitate to ask us in case of any further questions. Thank you.
Thanks so much, Sandra and Alvaro. That was a wonderful presentation. So this next part of the presentation is going to be presented by Lynn Santos from STEMCELL Technologies. And she's going to talk about compliance for clinical grade iPS lines. So I'm going to pass it over to Lynn now.
Thank you for joining our webinar series today. And my name is Lynn Santos, and I am the Senior Director of Quality Assurance and Regulatory Affairs at STEMCELL Technologies. Today, I wanted to go through some interesting clinical market trends. Also want to speak briefly to where quality considerations begin, as well as cover a compliance hot topic that is safety by design. So some interesting global market trends as outlined by the Alliance of Regenerative Medicine 2019 report, as well as clinicaltrials.gov, shows that the number of regenerative medicine companies worldwide is growing. And there are over 1,000 clinical trials underway, and approximately 50 of those trials are using iPSCs, which is definitely an increase over the last couple of years. In almost all major jurisdictions, they have a fast-track process in place for cellular therapies. And with that is excitement towards commercialization, but also some quality considerations. So when you increase the speed to market for, let's say, 10 plus years for a traditional drug product to 2+ years with the cellular therapy, you will arrive ready to commercialize your product and justify your readiness to your regulatory authorities with a much smaller data set. So with that comes risk that needs to be managed from the beginning during design and as well as in past commercialization. STEMCELL is proud to be a part in supporting over 45 clinical trials around the world using several of our different products. Now, where does quality begin? Sometimes people believe that you can test quality into the final product and this will suffice. But I found this very interesting quote from a recent paper that was published. It's called GMP Banking of Human Pluripotent Stem Cells. And I thought that the quote was quite poignant. It states that the quality of the final product is directly related to the quality of these seed materials used. Poor quality starting materials can directly impact downstream lineages, affecting safety and reproducibility, jeopardizing results and wasting resources. So this, in essence, is stating that you really must consider quality right at the beginning. And this begins with your starting materials as well as your raw materials. So there are 3 pillars that the regulators have proposed to the market to effectively support patient safety. And this has really been a compliance hot topic as we have gained feedback from our customers as well as regulators directly about their concern of the potential of transmitting pathogens. So how do we mitigate the risk of viral contamination into our products and then, of course, into our customers' final cellular therapies? So again, there are 3 pillars that are outlined and to assure patient safety. The first one is sourcing. So this is around where you get your raw materials. And the other 2 are testing and processing. But with those 2 pillars, there are limitations for cellular products. because for testing, it would be very difficult to determine which virus panels you should test for if you are unaware of what possible animal components were used upstream. Processing also has its limitations because the labelness of cellular products, they cannot be exposed to extreme processing measures such as high temperatures or high or low pHs. So therefore, you need to push that quality back into your design by creating controls for your raw materials. And to get a handle on exactly how deep you need to dig, I will walk you through this particular pyramid. So most of you as sponsors of clinical trials or manufacturers of iPSCs ready to be used in downstream applications are really thinking at this top level. So the raw material that we provide as a supplier, for example, from STEMCELL, is considered the primary level raw material. And often suppliers will provide a certificate of origin that only goes to the primary level. So you need to be aware of this and ensure that they also provide you with traceability down to the secondary level. And these are subcomponents that were used to manufacture the primary level raw material, and even down to the tertiary level, which are the subcomponents used to manufacture the secondary subcomponents. So to demonstrate this example, there are several suppliers out there that claim animal component-free cell culture media. And this is exciting that this is now being available to the market, but it is important to know that there is no standardization on those claims. So you, again, as a sponsor, are accountable to ensure that your final product has a viral safe profile. So in this example, you receive the material and you may receive a certificate of origin that highlights that no animal components were used at the primary level. Then if you have enough traceability on the certificate of origin, you will recognize that perhaps a recombinant protein was used. And although it was not sourced directly from animal materials, you will determine that there was porcine trypsin that was used as part of the manufacturing process for that recombinant protein. So now the regulators will expect that you will have documented evidence of the viral safe profile of that porcine trypsin. So I'm going to walk through the three pillars that I highlighted in an earlier slide. So the first one is around sourcing. So this is documented in a detailed certificate of origin. So as I alluded to earlier, you need to be aware to what level of traceability that certificate of origin is being provided. So if it is only to the primary level, you need to get back to your supplier and make sure that they can provide it to the secondary and tertiary levels. On that certificate of origin, it should be documented that any animal components were sourced from BSE, TSE negligible risk regions, that the material was sourced from low risk tissues, for example, blood versus the brain, and the mode of collection needs to be considered as well. Once you receive this material, you'll conduct a risk assessment of each raw material as it relates to your process. The second pillar is on testing. So this too is captured in a detailed certificate of origin. And for example, in the previous porcine trypsin that was used for the manufacturing the recombinant protein, so that would need to have evidence of adventitious agent testing for a porcine panel of viruses. You do not need to determine which viruses those are. The regulations will highlight what those are for you. The amount of testing and when testing occurs should be based on a viral risk assessment. Effective processing of raw materials is the third pillar. So, for example, if you know through your certificate of origin that the material contains BSA, then there should be documented evidence that the BSA had undergone a processing step that removes viral contaminants, such as being exposed to higher low pHs, higher low temperatures, chemical conditions, and nanofiltration. So processes should be validated to ensure consistent performance, and this will be demonstrated through viral spiking studies. So in summary, how can your supplier help you? First, they need to provide you with a detailed certificate of origin. This is a great place to start. Yes, you need the certificate of analysis as well, but to deal with this hot topic of a viral safe process, a viral safe product, it is important that you get that detailed certificate of origin. Sometimes the CDA may be required because there could be some intellectual property that may need to be shared. A quality agreement is a great tool to emphasize alignment between the two parties, and this is a requirement as part of GMP on the sponsor's part as well. Continuous communication is critical because, as we know, the regulatory landscape around regenerative medicine continues to change rapidly. So it's important to ensure that continuous communication occurs so that alignment of expectations can continue. Also, in the EU, STEMCELL, as well as other suppliers, can provide you with a regulatory support file. Sometimes you may not get enough information in the certificate of origin, and the regulators may have further questions, but we may not be able to share that with you because it is proprietary. So what we can do is we can provide that information directly to the regulatory agencies for their review in relation to your submission. So for more specific information on how we can help you qualify raw materials, please visit our website and search for ancillary materials. Thank you.